dear students of St. Peter's College, Colombo Gamma branch, to my lesson on grade 12 biology. Today my lesson is on plant tissue systems. When we consider plant tissue systems are originated by the meristems. Meristems are another plant tissue system which is capable of dividing. When new cells are arising from meristems, they undergo differentiation. So, meristems are differentiated to perform specialized functions and they become different types of cells in the plant tissue. So, these newly formed cells from meristems undergo changes in their cell walls, cytoplasm, nucleus, and many organelles of the cells. And so in the tissue, there are several types of tissue according to their function. Plant tissues are defined as a tissue consists of group of one or more cell types which carries out specialized functions. It means a particular tissue is differentiated to a particular function. In a plant body, like in the animal body, there are different types of functions occurring. For an example, in the plant leaf, there are different tissues. One is epidermis. It is a protective layer, so it protects the inner parts of the plant leaf. So it is differentiated. So we will consider those things later. So in the plant tissue systems, vascular plants are having three main types of plant tissue systems. Number one is dermal tissue system, number two is ground tissue system, and number three is vascular tissue system. If we go to the dermal tissue system, it is a protective layer of the plant. It is the outer protective covering of the plant. So this is a diagram of a plant leaf, TS of a plant leaf. So the cell layer in pink color is the dermal tissue system of the plant. So it is the epidermis. For an example, they have given the epidermis of the plant leaf, and epidermis can be there in young plants too. So it is the protective color. Here, top and bottom, both the epidermis layers are protecting the plant leaf. So, in the primary plant body, the dermal tissue system is existing as the epidermis. So, in this layer, other than the dermal cells, there can be some cells are differentiated into guard cells, trichomes, and root hairs in the young root. And epidermis is covered by the cuticle. Cuticle is a waxy layer. So, if we go to the trichomes, it is a type of cells in the dermal tissue system. It is an epidermal outgrowth. There are two types of trichomes, non-glandular trichomes and glandular trichomes. These non-glandular trichomes are also known as hair-like trichomes, and it reduces the water loss of the plant. And also, shiny hairs are existing in some plants. They reflect the excess sunlight when the plants are usually doing photosynthesis. Some trichomes are secreting chemicals. There are some chemical vesicle-like parts in the trichome. So, they are glandular trichomes. They secrete chemicals. it is a defense mechanism against the herbivores or some pathogens in the plant. And when we go to the ground tissue system, there are many types of cells in the ground tissue systems. The main three types of cells are Parenchyma cells, Collenchyma cells and sclenchyma cells. And there are specialized functions in the ground tissue system: storage, support, photosynthesis and short distance transport. So usually the ground tissue system fills the gaps in the plant. Between the other tissue systems, the ground tissue system, by the name it confirms, it fills the gap between the other tissue systems such as dermal tissue and the vascular tissue. So it is there in the plant body, mainly in the cortex and the pith. So these are the three types of tissues in the ground tissue system: parenchyma tissue, collenchyma tissue and sclenchyma tissue. So the next part is parenchyma cells in the ground tissue system. So these cells are living cells. We say these cells are living cells till the functional maturity. It means when the plant body undergo the secondary growth and these cells are remaining living, and these cells are having a primary cell wall. It is not a secondary cell wall. That's why the cells are living. And the primary cell wall is relatively thin. And when we go to the secondary growth, it is becoming thick. So this is a thin one because they are primary cell walls. And they have a large central vacuole here, and the cytoplasm has been gone to the periphery of the cell, and there are intercellular spaces between the cells in the tissue. So this is a specimen under the light microscope how the parenchyma cells look like. They have intercellular spaces and you can see the vacuole. There are many functions of parenchyma cells. Most of these cells are doing many metabolic functions. So sometimes they are secreting enzymes or some chemicals, various organic products they can be producing and secreting. And it is also a packaging tissue. As we mentioned, it is always filling the gaps. So it is as a packaging tissue, it may be in the cortex and the pith. And it is also doing storage. So when they do storage, they store starch. This is an electromicrograph of a storage cell after doing the iodine test. You can see as a result of the iodine test, the starch granules can be seen like purple or dark blue in color. So this is how it acting as a packaging tissue. In the pith you can see a lot of parenchyma cells can be seen with intercellular spaces and also as living cells. And these cells are still having the ability, like the meristematic cells, to divide and differentiate. They can be differentiated into other tissue systems according to the need of the plant. So this ability is also useful in wound repair because plants are always undergoing damages by the herbivores and various pathogens and environmental conditions, etc. So wound repairing ability is very important in this tissue system. And this ability is used in the tissue culture practices in the laboratory. So the second type of cells in the ground tissue system is Collenchyma cells. Collenchyma cells, by the name, the corners are thickened by cellulose. This diagram shows it correctly. The corners are, the cell wall is having cellulose and the corners are thickened by the cellulose, and compared to parenchyma cells, these cell walls are more thicker. This is a real electromyograph under the light micrograph. So it shows the Collenchyma cells, uh, and these cells are generally elongated, and they have thicker primary cell walls and intercellular spaces. It is may be present, we can't say absent, it may be present or less. It is not common as the previous one, the parenchyma, and this so these cells can be seen in young stems and petioles just below the epidermis, and with extracellulars the cell walls are thickened. Those are the characteristics of Collenchyma cells. Uh, and the function, if you go to the function of the Collenchyma, it is a mechanical tissue because it is having more strength. That's why they have stronger cell walls. So it gives mechanical support to the leaves and stems without restraining the growth. So it is a mechanical tissue. It is considered as the mechanical tissue. You can see it is there. Collenchyma cells are just below the epidermis. And here, this is a real diagram, so it may not be very clear. Here it is a line diagram with colors. You can see this is the epidermis, and Collenchyma cells are there with thickened cell walls under the epidermis. Our next type of cells in the ground tissue system is sclenchyma cells. Sclenchyma cell is having more thicker walls because now it is not cellulose. Other than the cellulose cell wall, lignin is deposited in the walls of the sclenchyma cells. So the cells are not living anymore. They are having some cytoplasmic connections known as desmodesmata, and simple pits are appearing in the wall, and these secondary walls are more thick, and that's why the pits are there. And the areas without lignin deposition in the secondary wall are known as plasmodesmata. Those are the connections with the neighboring cells, and they are dead cells at their functional maturity. So when we view through TS, you can see the shape, how the cell wall looks like, but really the cell is an elongated cell at the longitudinal view or LS. This is how it looks, but when you cut a TS, it is having this shape. There are types of sclerenchyma cells. The main two types are fibers and sclerates. Fibers are elongated and sclerates are usually known as stone cells, and these are circular or globular. The main function is the providing strength or specialized to provide the strength. You can see the lignin cell walls are there. Fiber, by the name, it is elongated, and this one is globular, the sclerate or the stone cell. The sclerates also, there are many types, many shapes are there. Most common shape is this globular shape and star shapes. There are many more shapes, but these two are most common. And these cells are shorter and wider than fibers, and it has an irregular shape. It also has thick lignified secondary cell walls, and they are found where the growth has stopped, not in dividing parts of the plant body. So they are present at the dividing, not in the dividing places where the growth has stopped, which are permanent tissues. So you can experience these cells in some of the fruits like pears and soursop, and also those are known as coarse fruits, and some seed coats and nut shells like peanuts. All these are sclerates. The fibers are the next type of sclerenchyma cells. They are grouped or they are occurring as strands. These are long, slender, and tapered. They are having tapered ends. And you can see the commercial fibers like coconut husk fiber, hemp fiber, xylems and phloems and jute fibers, which are using in commercially almost the fibers of sclenchyma. So the next one is the vascular tissue. So the vascular tissue is having some two parts, as you know, xylem and the phloem. The xylem and the phloem are existing in the vascular bundle. This is a vascular bundle. So in the vascular tissue, here the xylem as protoxylem and the metaxylem, and the phloem is there. If you use an LS, you can see it is existing like combined manner. So these are flowing directions of the xylem and the phloem. They are occurring in the vascular bundle. When we go to the xylem tissue is consisting vessel elements, tracheids, fibers, and parenchyma cells. So in the vascular tissue, the xylem is one of the main parts. So it is having these types of tissues. When you see from TS, you can see the cells in circular or hexagonal shape, but elongated nature you can see from a LS, and you can see xylem vessels are elongated. They are having perforation plates. These are vessel elements and tracheids and fibers. And also the parenchyma cells, which we discussed earlier, also may be present in the xylem tissue. And all these cells are there in angiosperms and some of the gymnosperms, not in all the vascular plants. The vessel elements. The vessel elements are in the angiosperms and some of the gymnosperms. These vessel elements are cylindrical and long. You can see they are like cylinders and they are very long because they are transporting water and minerals. According to their function, they are tube-like. So they are wider and shorter and have than the tracheids, but they have thinner walls. The secondary walls are here deposited and they are thickened by lignin. And these lignified secondary walls provide support when the water is transported to prevent the collapse under tension when water is transported. So these cells are having at the ends perforation plates. Perforation plates are interrupted by some pores, and these pores allow water and minerals to transport, and the other walls are also having pits. So some pits are there in the other walls. Xylem parenchyma is also another type of cells in the xylem tissue, and these are living because they are parenchyma, and it is similar to typical parenchyma. And in the xylem, what they do is they form a living link between the. It is through the wood it forms, and between the pith and the cortex they form a living link because other cells in the xylem may be dead. So this is xylem parenchyma in a LS. Okay, In this diagram, they show how Xylem parenchyma is existing with other xylem vessels and tracheates. Xylem fibers, as the previous fibers, they are elongated because they are originated by the usual tracheates, and they are shorter and narrower than usual tracheates, and they also have pits similar to the tracheates. They have thicker walls and narrow lumens, but they do not conduct water because they are not specialized to conduct water in the xylem. In the xylem, there are vessel elements to conduct water. The end walls are overlapped because they are existing as groups, and these are stronger and provide strength to the xylem. When you go to phloem tissue is differentiated to phloem translocation. It means transport of sucrose or produced food by photosynthesis throughout the plant body. They transport this unidirectionally and bidirectionally in both, because in the xylem they only can transport unidirectionally, and in the xylem there are some cells like other cells. So in the phloem also in the same manner, there are some cells, CO2 elements, companion cells, parenchyma cells and fibers. All these 4 types are present in the angiosperms, but in other vascular plants it may be present some of them. So the main function is phloem translocation, and also these cells are living except the fibers. Fibers are non-living. So this is the CO2 element and the companion cell, and the fiber is here, and the parenchyma. Here parenchyma is existing in the tissue as in the xylem. There are another type of cells, not CO2 element, Sieve cells. These are present in seedless vascular plants. This is a Sieve cell. It is not a CO2 element. This is the structure involved in translocation in seedless vascular plants which are lower and some gymnosperms. And in them, CO2 elements and companion cells are absent. So, anyway, they have to transport organic food. So what they do is using these Sieve cells. So the companion cells are non-conducting, and it is along with the CO2 element. So if we go to the CO2 element, it is a structure differentiated to transport organic food. So companion cells are non-conducting, but they are in the alongside of the CO2 element, and they are connected to the CO2 element with numerous plasmodesmata. So as a companion, they are helping the CO2 element because CO2 element is absent with the most important cellular organs. So companion cells help in phloem loading in leaves and in other organs, phloem unloading. Phloem parenchyma and fibers are also there in some of the plants. So what they do is they also found in dicot plants, and they are similar to typical parenchyma and generally elongated, and they occur in medullary rings and vertical strands and forming the living link as the xylem. So they are similar to sclenchyma, and they occur occasionally in the primary phloem but abundant in the secondary phloem. So they are forming vertically running bands, and help the phloem to resist the pressure when the phloem is subject to stretching when the growth is continued. So, this is my question. Differentiate the xylem and phloem. So, xylem and phloem are having some differences. So, mainly it conducts water and minerals, and phloem conducts organic solutes. So, xylem is unidirectional, while the phloem is bidirectional, and the conducting element is the tracheae and the vessels, and the phloem is having CO2 elements, and uh, some are dead in the xylem, some elements, tracheae, vessels and fibers, and in phloem most are living, especially the seal tubes, companion cells and the phloem parenchyma. Only the dead tissue is the phloem fibers, and xylem provides mechanical strength and there's no mechanical function in the phloem. So this is the end of my lesson, and we will meet with another type of plant form and functioning. Thank you and have a nice day.